for today, for everyone, what I'm going to talk about is we actually looked at, and by the way, no one else is doing this. That's what's unfortunate. The press ain't doing it. The academics aren't doing it. Why isn't someone giving us a critical analysis of hydroxychloroquine, remdesivir, or vitamin, D and C? So I'm going to go through that right now, and obviously, any one of you, I'm going to leave everyone on. If you have a question, just jump right in. And Jen, if you're listening, if you want to see a question you want to share with me from home, just text it to me, and I have my phone available. So let me just jump right into this. Let's go over here. Okay, here we go. So I have a set of slides. You guys can watch it on YouTube, and people out on Instagram. This is Dr. Shiva. I'm going to start really discussing, giving a science lesson, which I promised it was supposed to take place several hours ago, but I'm on Indian time. Sorry about that. So anyway, let me start over here. By the way, first of all, everyone, if you want, you can go to a site called vashiva.com, victoralphashiva.com. And if you're on Instagram, I'm streaming right on YouTube. I'm streaming on Facebook as well as Periscope. But if you go to vashiva.com, you'll see there's a lot of things I do, from creating the first email system to running for Senate and Cytosolve. Cytosolve is a 23rd century platform for discovery, but basically platform means it's a software company. We have the capability to model molecular pathways. And what Kennedy did, was, if you look below the fold, we have all different logos of different people who come to us at different points and wanted to work with our software, from doTERRA to Juice Plus. In fact, all of our companies, except the research institutions we work with, are all natural products companies. We worked with Pfizer many years ago, about 3 or 4 years ago. They spun off an innovation group, and they were very interested in the models that we had for autoimmune disease. And this was a radical group which recognized the pharmaceutical process was all screwed up. And we started with them, and then in the middle of it, they reneged. The 2 people who we started working with just disappeared because they had had enough of Pfizer. So that's what. But basically, we sell software. We create mathematical models, and I'll show you what that means. So.
Excuse me, Shiva.
Yes.
People can't hear you because Catherine Vitale is in the background.
Oh,
can you mute everybody?
Yeah, I can mute everyone. Is that better?
Thank you.
Okay. I think Catherine was. I'm not going to mute everyone if you want. Is that better? Thanks. Everyone can hear? Okay. Richard, you want to just take that? Take that thing out. Yeah, just pull it out. There. And you can rest it right here. Okay. So Richard is helping me here. Thanks, Richard. So what we do at Cytosolve is, our goal is to really revolutionize the discovery of medicine, be it foods and supplements and therapeutics, for the right person at the right time. And as I mentioned, it's really a technology for modeling biological functions. For example, the immune system on the computer, which we call in silico. And the goal is to discover different types of medicines faster, meaning so you can save people's lives, or validate if mixing together turmeric and resveratrol has any effect on you, and to eliminate the need for animal testing. And this is typically the process today in which big pharma works. Big pharma does this very interesting process where they take a new compound, they test it in a test tube, then they test it in animals, and if it makes it out of that, then they go through phase 1, phase 2, phase 3, what are called human trials. It takes around 13 to 15 years, some of you may know, and this whole process is quite expensive, but moreover, it's not that efficient, because after all those years, what comes out at the end could be really harmful to you. And it's not that different than when people used to fly airplanes in the old days, or make airplanes in the old days, or just create the wings. They would just make something and then throw a pilot in it, and if the pilot didn't crash, they were happy, and if he did, they said, gee whiz, that's what happened. So, and the reason that the entire pharma model is so screwed up is because the academic research model is so screwed up. And if the elephant is something, let's say like your cancer or some complex disease, the individual scientists touch pieces of it, and they actually don't look at the whole. So they don't look at the whole elephant. they're touching parts of it, and if they were ever work together, they wouldn't really find the whole. They'd find something like this, because they all have a blinded view of reality. So my goal was to figure out if there was a different way to do this, particularly given how screwed up the economics have become for pharma. Year over year over year, they spend a ton of money in R&D, and they're finding less and less new drugs. And so they spend a ton of money here, and you can see it here. And Venu, I think from your background, from a healthcare perspective, this has gotten pretty bad, right? What, 17, 18, 20% of our GDP is now spent on healthcare.
Oh yeah, it's going to keep going exponentially higher because of the setup of the system.
Yeah. And what is some of the key points of that, Venu? Why is the system so screwed up?
Well, it's the reductionist model that you just identified. If everybody's in silos, I mean, I'm a gastroenterologist, I'm a holistic gastroenterologist, but most of these fields are subspecialty fields. You hone in on things, and there's merit to that. But then when you lose the sight of the fact that a person is a whole person, and the ankle bone's connected to the foot bone, as you say, when you lose sight of that fact in Western modernist medicine, you lose sight of true health.
Yep, and we have a chiropractor here too. You want to say something, Tony, about that? Tony's right here. Tony, why don't you stand right there, because then we can get you on camera.
All right.
Yeah, so we also, we don't, chiropractors are very, very, you can stand right there, Tony, so I can just point the camera.
Sure.
Go over there, Tony. It's right there, so I can get you on camera. A little bit over.
Sorry. There you go.
There, so Tony, you're a chiropractor, right?
Yes.
You guys study the body as a whole, right?
Absolutely. That's one of the primary things that attracted to me when I discovered what you were all about. I said, this guy sounds like a chiropractor. He looks like a smarter version of me. I said, so I just got more and more into what you were talking about. And it's frustrating because our patients that I see all the time are so programmed. And it's just was very refreshing to hear your approach, because it just makes so much sense. It's just very common. I mean, I never immunized my children. I got 3 children now that are adults. I myself have been to a medical doctor for 36 years. Take care of myself. I'm a vegetarian. It's not that complicated. And if you look at the person as a whole, like you're talking about, it makes all the difference.
Yeah. And I think the thing that we talked about earlier was that the practitioners who actually still see the patients face to face are the ones who know, guys like you, guys like Venu.
We're on the ground.
You're on the ground.
We're dealing with people every day, and we're looking at the whole person and get to see, how they interact with the food and the water and all the chemicals they're exposed to. It's a complete picture.
Yeah. So thank you. Yeah. So I think the key thing is Venu's leading this whole effort for direct primary care, where the doctors literally basically become more entrepreneurial, and it's one-on-one, and get rid of the entire healthcare system. But pretty much our entire campaign and the kind of people it attracts are people like that who recognize that the entire system's busted and you need to take a different approach. And this is very, very different than just throwing money at it, which is like the Affordable Health Care Act, talking a lot of nonsense, which is what the Kennedys do, or in Massachusetts. They never really want to solve anything. It's all about how everything's positioned. It's all PR, PR, PR. But anyway, if we go back to this here, there's a fundamental problem, because these guys just keep throwing money at it, and now they're not finding enough new drugs. And this is why they want to go to vaccines, because vaccines, they don't have to go through the same regulatory process, and you can't sue them. So when I got into this, back into this, from the Western standpoint, one of the things that was happening was that when the Genome Project ended in 2003, as you can see here, it turns out we don't have 100,000 genes. We only have about 20,000 genes, the same number of genes as a worm. So that gave rise to a whole new field called systems biology. And systems biology basically said, if you want to understand the whole being, you have to go connect at the molecular level, the cellular level, tissue level, and you have to connect all those parts. And in 2003, there was a big idea that could you mathematically model the whole human cell. So if you think, and this is what the National Science Foundation put up, because they realized this was no longer, can you address what chiropractors, oops, I couldn't see that. Someone wanted, can you address why chiropractors and massage therapists are not covered under insurance? Is that in your bill? Yes, so we'll talk about that. Someone just had a question. But so in 2003, one of the big ideas was, could you mathematically model the whole cell? Because if you could do that, you could really start understanding very complex functions. If you think about the cell as this complex set of reactions that are all mixing together, imagine if you could model that. And to give people an idea, this is how complicated it could be, all these reactions. Now, this is I threw a couple slides in to do some education. So because Booby thinks we're a vaccine, I'm going to teach the difference between vaccines and a mathematical model. This is an example. If you remember basic chemistry from 8th or 9th grade, you have CH4 methane, which is a chemical, reacting with 2 parts of oxygen to get the product would be CO2 and 2H2O. Okay, so basically you take methane and oxygen, you'll get carbon dioxide and water. But this is what's called a chemical reaction. Now your body is composed of millions of tons of these trillions of chemical reactions that are occurring in real time. And if you could mathematically model all those reactions, you would have a predictive way to understand everything that's going on in your body. So that's what, and this is sort of the math. I don't want to confuse you, but just to let you know, what Cytosolve does, we do math. We make mathematical models. We don't make vaccines. So maybe that's what happened. He took a lot of heroin and he probably didn't understand the difference. That's what I'm assuming. Maybe he didn't understand the difference, Richard. What do you think? Between a vaccine and a model? That could happen. I don't know. But here's what we see, a mathematical model here. We see a chemical here going to another form of a chemical. And you can use mathematics, as you're seeing right here, to really understand this. It's called the Michelson-Menten equation. And this was a lot of what my thesis was based on. And based on the math, you could then literally draw a curve here, which explains how fast these chemicals will form. It's called a rate curve. But the bottom line is, if you have chemicals reacting together, physical chemistry, as it's known, gives us the ability to mathematically, predictively model this through physical chemistry. It's a beautiful science. If I didn't do engineering, I would have gone into physical chemistry. I probably wanted to do another PhD. It's an amazing field. But you could actually understand how things interact, and you can model it so accurately that the physical results match that. So imagine if you could now take more complex reactions and you could convert them to models, or even the complexity of this. So in order to do something this complex, people thought this was intractable. So the technology I created, the software I created, allows us to take these very complex conditions and literally convert them to systems of mathematical models, which people didn't think was possible. But I'm a software guy, and that's what I created. I created the first email system, created EchoMail, created many other systems, but this was a cool challenge. And the way we approached it, we said, if you take a complex molecular system, you have pieces of it, so don't try to solve the big, you're never going to do it. Decentralize it into smaller components, and then do what's called a coupled system of doing it. And that was Cytosolve. I'm not going to get into it, but this was really the technology. And then I wrote one of the first papers on it in 2011. It took me 3 years to publish this paper, because first time I submitted it, got rejected. People didn't understand it. Then we had to go to another journal, and then finally people got it. Because when you do something this radical, there's a problem in these stupid peer-reviewed journals, that your quote-unquote peers never want anything new coming. So anyway, we got it done, which was a feat. But as Venu said, he read my thesis, and we wrote many papers after, because in this world of science, you have to prove yourself. But anyway, the cool thing was we had this great article come out called Creating Accurate Models of Life, and Cytosolve was featured, and this is an IEEE, which is a very prestigious journal to be in. But the cool thing is, as anyone reads this, and any people out there who are confused, what is Cytosolve? We create mathematical models of life. And that's what it says, creating accurate models of life. But anyway, if you look at this systems process, what Cytosolve did was we figured out a way to take multiple different compounds like food, functional foods, supplements, even single compounds, which is the pharmaceutical companies do, and we license our technology where people can use this with our services to understand how that works relative to some reaction. And this is how we build airplanes, right? We don't simply throw pilots in and kill them. We do every airplane is designed on the computer. That's what Cytosolve is. So to say that, it would be basically saying a software company like a company that builds aircraft wings actually is a company that flies a plane around or makes airplanes. They don't. They basically create technologies which help you understand dynamics of events. And so we've created a whole powerful technology called Cytosolve. And what we're able to do is, in any field, we can take tons of papers. We just finished a project looking at 22,000 papers in the field of osteoarthritis, which I shared with people a few days ago. And we're able to extract the mechanisms, understand the pathways, and then figure out how foods and things work in that. And this was a paper that came out saying if you're going to solve cancer, that these are guys who are in the pharma industry saying if you're going to solve cancer, you can't use a single drug, you have to use combinations. And it's sort of that's what food is. Food is, if you think about it, food is medicine. It's a bunch of chemicals that come together. And my thesis was the only paper cited in there which could do this kind of mechanisms. And so in India, if you go, you'll have these yogis who mix together different kinds of chemicals, and here's what you're seeing in one of them. And mixing these chemicals they have figured out a way to help people heal different conditions. Now, if you ask them how did you come up with those mixtures, the Western scientists can't understand that. But with Cytosolve, we're able to literally bridge East and West. Here, what you're looking at is all the mechanisms of inflammation relative to a herb called curcumin or turmeric, which we've modeled, and we look at all the places, the red lines that herb interconnects within the cell. Similarly, we did that with resveratrol, as you can see, all the places, and then we have the ability to mathematically model it. So each row here is an experiment, and what you can see here is first experiment we're running with no curcumin, and we're creating inflammation in the body. That's what that 0.15 is. And then we just do curcumin, and then we just do resveratrol. Obviously it comes down, but what you can see here is when you do combinations, the same overall amount, that the right combinations can really bring down the level of inflammation. And this is really the notion of synergy. So let's talk about COVID now. So how many of you have really understood what goes on when a virus replicates? And this is what's going on. This diagram that comes, actually you can find it online, it's called Chemistry Views. This is not our diagram, so we thank them for that. But what you see here is this, think about the pink area here is really the inside the cell, and the stuff in the green area is outside of the cell. And what a virus tries to do, the purpose of a virus, or the virus is sort of interesting. It's not really a living organism. It's sort of an interesting creature. And I think there's a lot of research that still needs to be done on it. But a virus fundamentally tries to take over your cellular machinery and then replicate itself. So it basically wants to enter your cell and basically use your Xerox machine to make more of itself. So if you think about your cell having someone coming into your home, and they want to come into your home and then put themselves on your Xerox machine and make more copies of themselves. That's what a virus is trying to do, that simply put. And it sort of looks like a Xerox machine, in fact, when I show you this diagram. So a virus, the first phase it does is, those are these particles. So if you're looking at the SARS-CoV virus here, this is, the blue is the encasing, the red and the green there is what's called the ribonucleic acids, and the things that stick out of it are called the spike proteins, okay? So what happens here is this virus lands, and it tries to land on your cell I call it like the ball going into a catcher's mitt, and it's trying to land on the cell surface and try to find the right catcher's mitt where it can lock into. And that's called a receptor. In the case of this, it's called the ACE2 receptor. So the protein has to land right on it, and if it lands, a process called glycosylation takes place, and then that virus is able to get in to the cell. So it has to be able to glycosylate to get in. So right here, I'm going to make it a little bit bigger. Once it gets in, here's a cool picture. It gets in, the virus enters a cell body and releases its RNA. That's that green stuff. So it enters in and it releases its RNA. And that RNA goes into your cell Xerox machine, which are these ribosomes. So the virus comes in there, the RNA, and it literally starts replicating itself. And when it replicates itself, it's very fascinating because inside that RNA is the instructions on how it can make more of itself. It's quite cool. So it comes in and it uses your Xerox machine called the ribosome, and it starts this process. Now, one of the interesting things is the RNA actually makes, in order to replicate itself, it's got to make something called RNA-dependent RNA polymerase. And that molecule that it makes allows it to do replication of itself. So it's quite amazing. The RNA comes in, there's a piece of it which has the instructions to make more of it, okay? And then, so that's the first step. It's called RNA replication. So it's got to make all the RNA. And then the next process is it has to assemble itself back into the RNA, into the encasing, and then it has to transport itself out, right? So it's basically a 1, 2, 3, 4-step process. Enter, it's really 5. Enter, start releasing your RNA, replicating, 2, then build yourself, and then transport yourself out. So that's what happens, okay? Now, yeah, there you go. That's what happens. I think I did a bunch of these. Okay, now I'm going to cut to the chase. I'm going to tell you the end of the story. So what our analysis showed with cytosol, which is interesting, is if you go to the end of the story here. So for anyone joining new, this is Dr. Shiva, and we're literally going to cover vitamin, it's actually D and C, there's a slight error in the slide, and hydroxychloroquine. I'm going to change it while I have it right here. So there's two things we're looking at. We're looking at, actually, because it's a PDF, I can't do it right now, but I'll explain it. But here you're seeing it's not just D, it's D and C is the intervention, and that's hydroxychloroquine and remdesivir. So what I did was, if you look at those processes, one is you have the virus entry, you have the viral replication, you have the membrane disruption, and then you have to stop the viral activity, and you have to stop the cytokine storm. So there's multiple things that can happen here. You notice vitamin D actually kills the virus membrane protein. It's actually vitamin D and C. C actually stops the cytokine storm, which is where your body tries to attack itself, essentially. Hydroxychloroquine stops the viral entry because it stops glycosylation, it stops the viral replication, and it stops the virus particle assembly, and it stops the cytokine storm. So hydroxychloroquine is quite extraordinarily good, but you'll notice it has a lot of side effects. The vitamin D and C, this is, by the way, again, D and C. I wrote this wrong. And remdesivir does only one thing. It stops the viral replication. And we'll talk about that, and from a science standpoint, you'll see it's sort of limited because the virus can keep replicating. Now, from the side effect side, D and C have, frankly, no side effects. Hydroxychloroquine has about 60 side effects, and remdesivir, 75% of the patients who went through it had some type of side effect, and it's not fully tested. So you can see, when you look at this, you can try to, hydroxychloroquine hits 4 different processes, vitamin D and C hit 2, but remdesivir only hits 1, but the difference is D and C have no side effects. So we talked about this last time, that it's not the viruses that harm us, but it's an overreaction of a weakened immune system where it starts attacking itself. And so this typically happens if you have underlying pre-existing conditions, obesity, diabetes, etc. And this is another diagram looking at what they call the cytokine storm, when your own macrophages and T cells start spitting out a lot of cytokines because of a suppression of the immune system. Your own immune system, maybe the macrophages and the T cells are compromised, so you only have one system. So this is no different than if I took 2 of your hands and tied them behind your back and someone was coming at you and all you had was your legs to fight with. You'd probably flail like crazy and you could hurt yourself and others, etc. We talked about this last time when people do get, in this case, coronavirus, what ends up happening is here's a little piece of the alveolus, or an alveolus, we're looking at your lungs. It starts, because of the cytokine storm, starts filling up with fluids. You have all sorts of unfortunate reactions that are taking place here. Your body's trying to fight itself. Now, when they put you on a ventilator, unfortunately, you have more pressure coming down and it creates more damage. So the real issue is, should you be getting rid of this? And I talked earlier about the value of vitamin C in stopping the cytokine storm. So how does vitamin C work? So if you look at the mechanisms of how vitamin C works, it works in a couple of very interesting ways. Let me go down here. First of all, it inhibits, by the way, there's tons of research done on vitamin C, which I went through before. There's papers written where it says it can reduce the duration of mechanical ventilation. It can shorten the length of the stay in the ICU. It's supplementation. This is a big review that was done, has massive effects in again, lowering the time in the ICU. In fact, this one was in a very conservative journal. It's considered a big pharma journal, the JAMA, and they even concluded that vitamin C can reduce an amount of stay in the ICU in the most conservative case. But vitamin C does a couple things. It reduces what's called that cytokine storm, where it stops your body from going crazy. It immunomodulates itself, almost like putting shock absorbers on your car. And it has an antioxidant and an anti-inflammatory effect, and it has some antiviral effects. So here, this is how vitamin C does the immunomodulations. When your body is attacking itself, one of the interesting things people have found is there's a protein called GAPDH, or a molecule. It's overexpressed, which means there's a lot of it. So what vitamin C does, as you can see, it goes through this process in an interesting way. It creates these reactive oxygen species which go shut that down. Simply put, vitamin C has a very powerful way of making sure that your immune system stops overreacting. The second and third things vitamin C does, it goes in when your body's under this kind of assault, it creates a lot of reactive oxygen species, and vitamin C comes and shunts that, and it also lowers inflammation. So those are the things vitamin C does, and it has many, many other effects. But it's really, really profoundly valuable, especially when people are in that critically ill condition. That's why in the letter I wrote to President Trump, I recommended that you take high dose, especially in stress conditions. You wouldn't do that under normal conditions, but in stress conditions, it has a lot of value to really bring down that cytokine storm. Unfortunately, people like Fauci weren't really promoting this, which is sad, because you could have saved a lot of people's lives. In fact, they recently saved a doctor's life in Seattle who got COVID. He was going downhill, was put on a ventilator, and they, maybe because he was a doctor, there's more collegiality. They decided to help him, and they gave him a combination of an immunosuppressive drug and high dosage vitamin C, and they literally rescued him in days. So the other one is vitamin D. So my point is, if you combine D and C, how does that compare to the others? But vitamin D, nearly 80,000 papers written on it. A recent review article, over 11,000 participants, and they conclusively said across that review articles it was safe, it protected against respiratory tract infection, survival in ICU patients was increased, and it has greatest benefit for those people who are deficient. And it has a lot of different mechanisms of action. We've talked about, you have the innate immune system and you have the adaptive. You guys can go to truthfreedomhealth.com. I discuss all of that. There's a bunch of great videos there. But you have the innate and the adaptive. And if you look at that, the innate and the adaptive systems work together, but one of the subcell systems in the innate system is called the macrophages. And the macrophages are the ones that actually go destroy the virus replication process. And here, if you look at, if you're breathing and air is coming down, there's structures called alveoli, which is where gas exchange takes place. Oxygen comes in, CO2 goes out, and your blood gets replenished with great oxygen. And if a virus comes in, what happens? So your airways need to get protected. So one of the ways that this can happen is with the use of vitamin D, and vitamin D literally creates a chemical called cathelicidins. So I'm going to walk you through this. So here's one of those alveolus, and here's these blue structures are your macrophages, and they're trying to kill this virus here. And how do they do it? Well, there's two types of macrophages: anti-inflammatory, which is not what we're going to talk about because they don't really have the effect here. It's really the pro-inflammatory ones. And here's a wonderful paper that really talks about, I'm going to walk you through this here, that your body actually, by the way, this mechanism for bacteria as well as viruses, when a bacteria comes in or a virus comes in, your body will literally the macrophages need to go take out that enemy. So vitamin D comes in, and vitamin D literally through a set of processes allows the capture of that pathogen and then the destruction of it. And the way it does it is through the creation of these chemicals called cathelicidin antimicrobial particles. So CAMPs. It's like you're going to summer camp, but it's called cathelicidin antimicrobial particles. And the way this works is when vitamin D comes in to your body, could come in from as a result of sunlight, your body creates it, or you take it like D3 cholecalciferol. And when you take these vitamins, what the body does, it literally converts that vitamin D3, it converts vitamin D3 through a series of processes you can see here to produce these chemicals over here called cathelicidins. And if vitamin D3 is a gun, cathelicidins are like bullets that literally go shoot, they go capture, and then they break down the walls of the bacterium or the virus. And here we're looking at the HIV virus. But the cathelicidins help this process of capturing the virus, and then when it's in there, literally breaking down the cell walls. And I'll show you this. And by the way, here we're looking at bacterium, same thing. And so this is a wonderful paper that came out in Nature in March of 2019, and it looked at 3 types of cathelicidins. Again, the cathelicidins, to be clear, are the chemicals that your body creates with the presence of vitamin D3. If you don't have vitamin D3, you're not going to be able to produce those cathelicidins. That's why sunlight is important, because when you get sun, UVB radiation hits you, you create vitamin D3, and then your body upregulates these cathelicidins. And so here in this paper, they looked at 3 types of cathelicidins. Porcine, which means pig, which is PMAP36. It's a certain type of cathelicidin. LL37 is what humans have, and Cath2 is what happens in chickens. And what they did here was, it's a cool article because it says almost all cathelicidins show in simple media direct antimicrobial activity against many different bacteria, viruses, fungi, and parasites. So think about that. This is the importance of the sun. You get sunlight, your body, by the way, dark people need more sunlight. People who are dark are actually more prone to reduce vitamin D3. So let me explain why a lot of the African Americans were having more problems with quote unquote COVID or flu. But what you see here is against bacteria, viruses, and parasites. So here's sort of a chemical way of looking at it. This one is the pig cathelicidin, this is the chicken, and here is the human. But what you can see on this graph here is as you increase the volume of cathelicidins, bacterial levels go down. By the way, this is also true for virus too. And this is how these cathelicidins work. They literally go to the bacteria or pathogen, and they surround the cell wall of that bacteria. That's what those little daggers are, which are the cathelicidins. They take over the membrane, and they literally dissolve it or burst it open. It's almost like a soap inside your body. And here's another example of it. And I wanted to show you these. This is literally, microscope pictures where the far left is a bacteria. This is the pig. This is one with the pig cathelicidins, and one with the chicken and the humans. Since we're humans, you can see here's a cell surface, and you can see how the end cell walls are busting up here. See that? If you want to look closer. So you can see the power of these cathelicidins to literally break up the wall. So vitamin D3 creates cathelicidins, which literally is an antimicrobial. So Here's a summary on vitamin D. You get it from sunlight, dietary supplements, fatty fish. It's about a $1.7 billion industry by 2025. However, that's very different than the vaccine industry, which is nearly $60 billion, and is going to double to about $120 billion in 2026. So you're looking at a $1.7 billion industry, vitamin D3, pennies, and you're looking at a industry which is, 660 times that size. So basically, in closing, vitamin D and C together, one really is an antimicrobial vitamin D, and then vitamin C, vitamin C has many different effects in terms of really bringing down inflammation, modeling it, reducing the cytokine storm. So let's look at hydroxychloroquine. So hydroxychloroquine, one of the most important things, remember we talked about the viruses has to get in. So hydroxychloroquine literally stops what's called the glycosylation process. When that virus comes and it tries to connect to that AC2 receptor, it actually stops that. So that's a pretty strong capability of hydroxychloroquine. The other thing hydroxychloroquine does is it actually stops viral replication, and it stops viral assembly, and it stops the transport. It affects 3 different things here, as we talked about. The other thing hydroxychloroquine has, it has some effects on immunomodulation, which means bringing down that cytokine storm, and it also has anti-inflammatory effects. So in many ways, it's awesome, but it has a lot of side effects. I've listed about 60 here. So a lot of different side effects. Now, people also say you should take it with zinc. Zinc is something that really supports the inhibition process at that ACE2 receptor. So it sounds good, and I think this is why the president really liked it, or his advisor, somebody told him. Fauci obviously didn't. Fauci wanted drugs. Little guy. That's what he wants. Sorry about that. And you can see Sanofi's stock, which was tanking, got a really nice boost from all the stuff that went on there. Let's go to remdesivir. This is sort of interesting. So I'm going to walk you through the latest clinical trial that just occurred. Gilead Sciences is a company that created this. By the way, that organization received gave a lot, about $250,000 to the Clinton Foundation, and you can. I did a tweet on this, and their stock since this came out has gone up by 40% over literally the last about 60 to 80 days, 36% to be exact. But here is Gilead Sciences. Their objective was to determine the optimal duration of treatment with remdesivir. So if you look at the clinical study, they weren't even comparing with control, which means giving some people the drug and others people no drug. They were just trying to say, how long should we give it for? So one treatment they gave people for 5 days, and the other people they gave for 10 days. By the way, do you guys have any questions over here? Is everything all right? Okay. So they basically were not this was a weird clinical trial. It's not like they gave one people the drug and then the other people didn't. They basically, everyone got it. Half of the people got it for 5 days, and the other half of the people got it for 10 days. So I hope that's clear. So they were just looking at how long the duration of the treatment would be. And the regimen, as you can see here, was that they gave 200 milligrams on the first day, followed by 100 milligrams each day, and it was done intravenously. So 200 milligrams on the first day, and then they followed up with 100 milligrams each other day. They had a total of close to 400, 200 in one group, which got the 5-day, and almost 200 in the other group, about 197 in the 10-day treatment group. Okay? So basically they split, 200 people got it for 5 days, and 197 people got 10 days. Okay? Any questions on that? Yes? Okay. Then this is what the patient selection criteria. So one of the things in these clinical trials, you have to say who are you going to test it with? Are you going to give it? Now remember, people are coming into the hospital. Some people are sort of okay, they just have a cough. Other people actually have pneumonia. and other people in critical condition, their lungs are filling up. So what they did here was they actually looked at hospitalized patients with severe manifestations of COVID-19, and the severity was to have evidence of pneumonia and reduced oxygen levels. That these were people, I don't know if you guys know, they didn't have to go on ventilators yet. They essentially had reduced oxygen levels. Let me just go to our group over here if there's any questions. Anyone have any questions over here?
Yeah, somebody's asking about interferon 2B, to do an analysis on interferon 2B.
Interferon beta? Yeah, interferon beta. Yeah, I've done. I've done that before. We could do a whole thing just on that. Tell them that I've done that. Part of my thesis was on that. But yeah. So any other questions? If you guys have questions, just jump right in. So they basically the patients that they selected for this was they had pneumonia. They're not critical, but it's like before ventilators, but they need oxygen. And patients on ventilators were excluded, as it says. On the clinical outcome, the endpoint was, so whenever they do a clinical trial, they say, when is it successful? That means the patients no longer required oxygen. That was one endpoint of success. Let me go back here. And the other endpoint of success was they no longer required medical care or they were discharged from the hospital, okay? So that was the endpoint of success. And what is pneumonia? Pneumonia is interesting. So what happens when you get pneumonia? So these people had to have pneumonia. And so when pneumonia it's really an infection, that inflames the air sacs, those little alveoli in one or both lungs. And the air sacs, start to fill up with fluid or pus. You start coughing, a phlegm, if people are just throwing up. I don't know, one of our volunteers was saying she probably thought she had it. I think about 3 months ago, I think a lot of people are getting this. We remember 3 of our volunteers. Remember, Karina? Like Elena and all these people. They were throwing up so much garbage. I think they were getting it. Fevers and chills. And this can be caused by microorganisms such as bacteria, viruses, or fungi. So pneumonia can be caused by multiple things, not just bacteria. And in acute respiratory distress syndrome is you have shortness of breath, you have low blood pressure, you have the thickening, and you have severe pneumonia. So there's pneumonia, and then this is essentially a much more severe form of it. So what is remdesivir? This is if you're interested, this is a chemical structure of it. If you understand some organic chemistry, but it's a chemical structure of remdesivir. And it's considered a broad spectrum antiviral for several viruses, including coronaviruses. And remdesivir, the actual thing is remdesivir triphosphate, RDVPTP, and it's a synthetic analog of ATP. This is what's interesting, adenine triphosphate, okay? And its efficacy against viruses is in decreasing order. So Ebola, MERS, SARS-CoV, and SARS-CoV-2. So they've used it against various things. So how does remdesivir work? This is sort of a cool diagram. So it enters remember the virus enters your cell through this ACE2 receptor, and then it goes through this thing called endocytosis. And in that process, the virus, remember, starts trying to replicate itself. And so this is what's called a viral RNA. And one of the important things is that the virus replication process actually creates this protein, which we talked about, called RNA-dependent RNA polymerase. Simply put, the virus is trying to replicate itself, and it creates this particular protein. And what happens is the remdesivir does something fascinating. It actually goes and mimics one of the adenine. Sorry, I have it right here if you can see it here. So RTVP inhibits the viral enzyme RNA-dependent polymerase, which is necessary. And what it does, it literally mimics one of the base pairs. And so what happens is, inside the cell, this is sort of cool. What happens is when things start replicating, you're starting to replicate your own DNA, or you're trying to replicate an RNA, there's actually other proteins which are making sure you're building the process correctly, and they try to fix it. But what they're doing with remdesivir is they're actually putting in a bogus base pair, and they're hoping that your own body doesn't catch that problem fast enough, the reaction rate. So this is interesting because this is a problem with remdesivir, in my view, that if that virus mutates, this drug is going to be useless. You have to create another version of it. So this is good under certain conditions, assuming the reaction rate of your body trying to fix itself, the virus, is slower than remdesivir's reaction in duping the body. So what remdesivir is doing is actually duping the body's viral replication process so your body can't fix itself. So if you look at this, it basically is affecting one part of that very complex viral replication. And if that virus mutates, it's frankly useless, okay? Because it has to particularly affect that one base pair. So in what they found out in the clinical trials was the early intervention with remdesivir led to better clinical outcome. Well, this is true of, even hydroxychloroquine, right? You get it early, it's better. You take care of your body early, but here with vitamin D and A, as I've talked about. So the early intervention with remdesivir led to better clinical outcomes. So patients treated with remdesivir within 10 days of symptoms onset which is 62%. So people who had it within 10 days had 62% improved outcomes. Let me show you right here. Compared with the 49% treated with more than 10 days. Okay, so it's fascinating. So within 10 days, you had a better result, improved outcomes compared with 49% treated after more than 10 days. So patients receiving a 5-day treatment course of remdesivir achieved similar improvement in clinical status compared with those taking a 10-day treatment. So here was a 5-day treatment group. The time to clinical improvement for 50% of patients was 10 days. 60% were discharged hospital by day 14. 64% achieved clinical recovery. 8% mortality rate, which means 8%, which would be 200 people in and 16 people died. 64% achieved clinical recovery. 60% discharged from the hospital day 14. The 10-day treatment group, time to clinical improvement for 50% was 11 days. 53% were discharged within 14 days. 64% achieved clinical recovery, and yet 11% there was a mortality rate. So this is the data. By the way, you can look it up. It's all out there. In fact, they put it as a press release on Gilead's website. So now the adverse effect from the 5-day treatment. So the safety was the following: 71% of the people had some adverse side effects. 71% in 5-day group. Side effects included nausea, acute respiratory failure, 6%, higher liver enzyme indicating liver dysfunction, 7.3%. The 10-day group, 74%, so a little 3% more, nausea, acute respiratory failure, and high liver enzyme. So in closing, what you see is you have vitamin D and vitamin C. It hits 2 of those points. Hydroxychloroquine hits 4 of the 5 areas, and then remdesivir hits only 1. And again, the safety profiles are there. So in my opinion, hydroxychloroquine with the zinc can have very positive effects. However, it has all the side effects, to about 60 of them. Vitamin D and A hit 2 of the points, no side effects, but they're hitting 2 critical points, modulating the immune system, as well as having antimicrobial activity. And then obviously this one, in my view, is very, very, it's a stopgap. It's on a scale of 0 to 10, it's probably around a 3 of a solution. So we're going to take some questions. Let's go over here. Let's take some questions from the team here. What do you guys think? Was this valuable, or was it totally confusing?
I'm going to highlight some things there that are important for me. So this whole issue of vitamin D, I mean, if you want to know what vitamin D is doing, you might investigate what's the correlation of vitamin D levels to intensity of COVID-19 symptoms. Is anybody doing that? Haven't heard about it. So, a lot of this gets down to, what is your worldview of what you think science is? And right now, what we have a lot of is scientism, which is, like a religion, which has a priestly caste with so-called expert scientists who are going to focus on remdesivir. It's not that remdesivir shouldn't be studied, it should be studied, but there's this overemphasis of focusing on that rather than asking some basic questions about what's the correlation of vitamin D and symptoms of COVID-19. That's one point. Another point is, when you have somebody who gets raided by the FBI for giving intravenous vitamin C for something that doesn't otherwise have a standard of care, that's completely warped at that point. It's like raiding somebody for giving zinc for a cold. We don't have alternative strong treatments for a cold, so why are you raiding somebody because they're trying to give somebody vitamin C? It's just ridiculous to me. And again, it starts to make this whole notion of top-down scientism medicine become a reality. And then getting back to Cytosolve, one of the things that I was really fascinating on Cytosolve from the beginning is putting different things together in a mechanistic way, but then in a systems way. So for example, a parent would want to know if you give a vaccine with adjuvants and let's say the baby's born by C-section or doesn't have any breast milk, has a warped microbiome, which I'm very interested in as a gastroenterologist, what would a vaccine do in that scenario? Well, if you just focus on a vaccine and you don't know anything else but that, you're not going to know that answer. So knowing these second and third order effects is what makes Cytosolve Cytosolve. So in that sense, it's not a vaccine maker, it's a vaccine truth teller. And that's what we want as parents.
Yeah, and yeah, it's basically a powerful technology that it's a scientific source of truth. We, for example, on the Cytosolve side, we work with a lot of innovative entrepreneurs. We found this guy who, in horses, he had found when you gave these two very interesting natural products, one comes from citrus and the other comes from a green vegetable, that it had an amazing effect. He showed me pictures of horses' legs this big, completely coming down, within less than a half an hour. Better than Advil, better than Tylenol, all these things. So we ran that through Cytosolve and we found that it hit 5 different mechanisms. We were able to validate it. We filed for some cool IP protection because what's going on now is Whole Foods basically watches what supplements sell and they just replicate it. And they're basically putting everyone out of business. So with Cytosolve, we're also able to help small entrepreneurs get patent protection for their discoveries. And we're basically going to be offering that pretty soon. But it's something that we're not making the product, but we are discovering what combinations work. We more recently were funded by a family foundation who was actually very, very concerned about young kids on Adderall and Ritalin. Again, these are drugs. And I'm sure the Kennedys Harvard Law School, by the way, a woman I knew many years ago, she said nearly 70% of the people there are on Adderall. And my nephew, when he went to elementary school, he said nearly 40, 50, 60% of the kids are put on Ritalin. It's quite amazing. And these are pharmaceutical drugs. So what we did was this foundation was very interested in seeing where there are alternatives. So we went through about 162 natural products using Cytosolve. And what we discovered was, Richard, this has gone out, if you can plug this in. We found out close to 162 natural products, which had some profound effects had some effect on, you have to wait, had some effect on brain health, but out of that we found 12 which actually had some really good effects. And then we, using Cytosolve, all the math, we combined them and we found some which had really, really good effects for short-term memory, long-term memory, as well as cognition. And those things, we as innovators have figured out how to put them together. That's what we do. We use math. We use basic physical chemistry to figure this out. Any other questions? But was this helpful? Does this make sense? So you have this very complex manufacturing process. That's what it is. Virus comes in, it's manufacturing itself, and remdesivir throws a wrench in one of those processes. But the virus can figure out how to circumvent that. So then you have to throw another wrench. But if we boost the immune system, which is vitamin D and C and A, you're basically shielding it so the virus can't even get into your factory. That's what you want to do. But the viruses are interesting. They enter, and then they're using your own body. Basically, it's a thief enters your home, like I said, and he's replicating himself by stealing your own Xerox machine. That's what a virus is doing. It's basically using itself. It's creating an environment where it uses itself to make more of itself. If you can just do it from here, Richard. Yeah, we're almost done. Yeah. Any other questions? And then hydroxychloroquine, this is what, it is powerful, but it has a lot of side effects. But with something like Cytosolve, had we had a chance to run all that through, we could have figured out what side effects. We also have the ability to load up people's genes, genetics. So you could find out, well, for this person, it's okay, and for this person may not be okay. One of the things that we just found out 3 days ago, this took a paper that I had worked on with a team at the military, a team at the U.S. Pharmacopoeia, which is it's a company that handles all sorts of dietary supplements. And we literally use Cytosolve to understand toxicity. I did this project 5 years ago. By the way, it takes so long to publish stuff because of all the politics you have to go through. But we finally got this paper published, but it validates Cytosolve's use to understand toxicities. I can bring it up if you guys are interested, but it just came out a few days ago. Anything else? Anyway, to all of you listening out there, this is Dr. Shiva. I want to thank Dr. Julapali, Venu, Debbie, everyone out here who stayed. We're going to end our marathon. It's getting late here. Unless people want me to keep going, I can if you want. We have Catherine Vitale, Shannon, Jacqueline, Karina, Debbie, Elizabeth. Who else do we have? And a couple of other people are coming and going. But we have a bunch of volunteers. We have thousands of volunteers growing on the ground, and our campaign is really about truth, freedom, and health. And I want to encourage everyone to take a little bit of time and go to our website. If you can go to shivaforsenate.com, SHIVA for Senate.com, you will see a great website that our teams put together. One of the most important things is we're going against the Kennedys, and the Kennedys are running a smear campaign against me because they have their little boy Joe Kennedy that I'm running against, who, by the way, already has $51 million in the bank at the age of 37, 38, when he's never really worked at all except be a congressman and go from Harvard and got a cushy DA job. We're running for Senate because we believe it's time that, working people like us, not the 0.001%, got in. So it's really about no to the Kennedys aristocracy. It's about yes to a people's movement, and that people's movement has a slogan: truth, freedom, and health, which means without freedom we can't get truth, freedom of discussion. Without truth and science we can't get to health. As Venu just said, we're actually practicing scientism, meaning it's like science by the majority or truth by the majority. Basically they think they can just lie enough. Like you're just seeing this, you lie enough that climate change is taking place, meaning CO2 is a pollutant. You lie enough that we all need vaccines. You lie enough that I'm a vaccine maker. You lie enough that I work with Bill Gates, and that's how you push things through. And interesting enough, the people doing this are the liberal aristocracy. You have Bobby Kennedy lying to people that he thinks CO2 is a pollutant, and he hangs around with Jane Fonda, gets some pictures with her, and thinks he's somebody. But he's the same guy who tries to push through lies about myself and others. And that's how they work. They work through just blatant lying.